Alrighty, I'm going to go ahead and start the recording now. And starting in 5, 4, 3, 2.
Hi, thank you very much for joining this session today. I'm Nandini Krishnamurthy. I lead the applications development for Inscripta. I'm very excited to talk to you about Inscripta's cutting-edge Onyx platform. Biology has unlimited potential to positively change the world. We firmly believe this at Inscripta, and many of you are harnessing the power of biology daily as part of your work. And what we want to do is build better tools for you that will accelerate your success. So we've developed the Onyx platform, a cohesive ecosystem that has software, instrument, consumables, assays that scales the microbial genome engineering. This platform helps you make custom libraries of genome-edited cells with push-button simplicity. In this talk, I will cover how the technology enables you and what applications you can use it for. We all appreciate the powerful foundations that drive biology. One, the ability to detect phenotypic variation, selecting for preferred phenotypes, and the identification of genotypes that cause the phenotypic variation. For centuries, biologists relied on evolution by sampling natural variation in populations and making inferences of genotype to phenotype relationships. The power of this approach is demonstrated in modern agriculture, of course, with both breeding of crops and livestock. We've been using mutagenesis to create variation, and that has led to a lot of discoveries in several model organisms. And more recently, we do have seen advances in directed evolution of proteins that even got Frances Arnold her Nobel Prize. But as powerful as these approaches are, they have a few limitations. For instance, once an interesting phenotype is identified, figuring out the genotype that actually causes this phenotype from the genome is pretty daunting. And there is no control over the type or location of this variation. And these limitations mean that there are very long cycles of learning. Many of these limitations are driven by deficiencies in our technology. While it is commonplace to be able to create variation across the genome, even with precise genome editing technologies like CRISPR, one is typically limited by the type of variation that can be created. For instance, predominantly what we create are genome-wide knockout libraries. How do we open up other regulatory aspects in biology? Conversely, when making a large variety of edit types, we are usually limited in the number of genomic locations accessible in an experiment. So this is what we show in the X and Y axis here. And to this, you can add additional challenges like slow, laborious workflows and long cycles of learning. So to truly advance biology, we need to get into this upper right quadrant. That is, we need to be able to generate a variety of edit types across the genome with speed, reliability, and ease. What if you could create a custom library of precisely edited cells within weeks in your lab with almost negligible hands-on time? This is what the Onyx platform enables. The Onyx platform provides the ability to create and track a wide variety of precise edit types across the genome. And this current version of the Onyx platform has been developed for E. coli and for Saccharomyces cerevisiae. The Onyx platform is designed in such a way to enable you to make libraries of variants with extreme ease. So let us look at what this platform provides. When you purchase this platform, you get access to the benchtop instrument and access to the software and consumables. The platform enables all steps of a DBTL cycle. For instance, you start with software that's extremely easy to design hundreds of thousands of edits. And then we have custom reagents and the benchtop instrument that enables you to create a genome-scale engineered library of cells by managing the genome editing and cell handling processes. Once you have the libraries of cells, we also have a suite of assays that you can bring together with your phenotyping of the libraries so that this enables you very rapid genotyping. As I said before, these are trackable libraries, so all the cells here contain a barcode. And the barcode can be sequenced through NGS sequencing that enables you to quickly identify the genotype that is related to your phenotype of interest. And this process completes with our resolver software, which enables you to identify the genotype and connect it to the phenotype. So by this combination of software, instrument, and analytics, we are able to create libraries of engineered cells which are precise, single edit per cell, diversity of edit types such as deletions, insertions, and substitutions, and enable rapid genotyping. So we spoke about scale, performance, and ease of use earlier. So let's unpack that a little bit and see how the Onyx platform achieves this. So there are many aspects of this system that improves the overall workflow to make it extremely easy. Let's focus on scale. Traditional approaches to scaling up genome engineering typically involve use of microtiter plates and delivery of reagents into individual wells to keep them partitioned. However, our co-founder, Andrew Gast, found a very nifty solution to achieve scale that will help us move away from the traditional workflows. By covalently linking the guide RNA to the donor template, we have removed the need for physical partitions and can now use cells as the partition. And in addition to the guide RNA and the donor, we've also added a barcode to this construct, allowing us now to scale from hundreds of wells to hundreds of thousands of cells, which are also trackable. And this addition of the barcode enables rapid NGS-based tracking, which is NGS sequencing is extremely predominant now and very easy to accomplish. So the second problem that you usually face when you're scaling up genome editing is consistent, reliable performance. A big challenge is the nonlinearity of biological processes that results in very poor coverage of designs or a very low fraction of edited cells in the library. To overcome this, we have developed several transformational microfluidics devices that help ensure that you have consistency within the process. And this manages the nonlinearity of biological processes inherent to any genome editing workflow. So what this gives you is a very uniform coverage of all your designs and high fraction of the designs observed within your library. The ease of use and the performance is also enhanced by our very unique editing reagents that consist of all components that are required to deliver a successful editing reaction, including a guide RNA, a donor template, repair proteins. We also have proprietary MAD7 nuclease, which is of the Cas12a family, and we are at the barcode. So this is a complete solution, and it is episomal, so it does not require any background modification of your cells to say integrating the nuclease or the barcodes. And what this now, so we looked at scale and performance, and I will give you an example of how we enable diversity of edit types. We enable you to go beyond just knocking out genes. You can design a diversity of edit types that gives you control over biological processes, be it transcription, translation, or protein function. And here are a few examples of the variety of edits that we've demonstrated. The capacity to deliver up to 63 base pairs in any edit type enables you to drive gene expression in E. coli, for instance, by inserting a promoter RBS sequence in front of every single gene of E. coli. If you require, let's say, a more subtle control, you can swap a terminator sequence, for instance, and this is an example we've shown in yeast. You can also modify protein function by accessing coding regions by either, let's say, knocking out protein function by truncating proteins, by insertion of a stop codons, or modify protein function by scanning either selected sites or across the entire protein through saturation mutagenesis. So now, given all the diverse edit types that you can achieve on this platform, how can you think about library design strategies? So here are a few examples. We've worked on these strategies both internally and with collaborators, such as our early access partners. And I can show you here our libraries where, for instance, you have a genome-wide promoter ladder where we've taken 5 different promoters from iGEM of varying expression levels and inserted this into upstream of every single gene of E. coli. So you have a ladder of expression now that you can compare across the entire genome. Another example of gene regulation is our collaboration with Dr. Michael Brent at Washington University, where we have edited transcription factor binding site in upstream of selected targets. These substitutions then enable us to study the impact of swapping out transcription factor binding sites in combination with the targets that we selected. If you're interested more in, say, pathway optimization, we also have an example of a lysine pathway that I will talk to you more about in the upcoming slides. Here we've taken the example that I have here is enzymes of the lysine biosynthesis pathway in E. coli, and we have done saturation mutagenesis of all the enzymes, and we also use transcription factors that regulate this particular pathway. So this gives you a handle of how to study pathway optimization. We've also have a collaboration with Dr. Jeff Boeke at NYU, where we studied the rapid identification of causal mutation in an edited, highly engineered strain to identify edits that can revert the growth phenotype in the engineered strain. And I'll give you an example of this as an application. Several of you are probably interested also in engineering heterologous proteins or pathways that you have inserted into the genome of, say, yeast or E. coli. Our platform enables you to target the heterologous elements that you've introduced. So now what you have is a fantastic handle to both edit the native genome and the heterologous pathways that you've inserted. And we have several collaborations where we are studying that aspect as well. The library sizes can range anywhere from 100 designs to say 6,000 designs in yeast and 10,000 designs in E. coli. So this gives you a wide range of flexibility now to study libraries of different sizes and asking questions around the native genome or heterologous pathways of proteins that you've inserted, and across coding and non-coding regions where you can study biology, whether it be gene regulation or protein function modification. This flexibility and the ease of use and the scale-up is what really drives and accelerates biology. So let's look at an example. The first example I will talk about is the use of this platform in forward engineering to rapidly identify phenotypes of interest. And a second example would be in genome discovery, where you rapidly identify causal genes that control growth in engineered yeast strains. For the first example around forward engineering, we decided to engineer the native genome of E. coli to test how we could achieve increased production of high-value amino acid lysine in E. coli. Traditional approaches usually would start with only the selected targets that are known to be associated with lysine biology, and mostly focusing on proteins and trying to identify modifications of protein function. What we wanted to do was to ask the question whether we can combine two different approaches: the traditional approach, which is a very rational approach with known targets, with a completely unbiased approach where you target the entire genome with a wide variety of edit types. So for the rational approach, we identified the enzymes in the lysine biosynthesis pathway. We also identified transcriptional regulators of this particular pathway and built libraries that primarily involved saturation mutagenesis as a strategy. And for the unbiased approach, we went across the genome and created protein truncation using stop codon insertion across every single gene, targeting every single gene in E. coli. Similarly, we also built 5 different libraries of genome-wide promoter insertion, and each of these libraries included a promoter that I spoke about earlier from the iGEM set of 5, where they had an increasing range of expression that has been tested before. So now we have very valuable libraries that are regulating both protein function and gene expression. We built around 24 libraries, and this included library sizes ranging anywhere from 4,000 to 10,000 designs per library. That gave us about 200,000 designs in total. What we then did was to use a high throughput screening workflow. The output of the Onyx platform is a tube of cells that heterogeneous mix of edited and unedited cells. We then singulated these cells in a high throughput workflow and measured titer of lysine using a rapid fire mass spec approach. The other principle that we followed here was the idea of forward engineering being the rapid iteration through the phenotypic landscape. So we did not go really deep into the 200,000 design, and we only screened for 18,000 isolates. So it was a very shallow sampling of the set of designs that we had created. The graph here shows you results from the primary screen of this particular approach. And what you see on the x-axis are the different libraries, the center panel being the traditional approach that starts with known targets, which are all the pathway enzymes here. And it is bookended by the knockout and the promoter ladder libraries. The y-axis plots the fold improvement in lysine production compared to wild type. You can see from primary screening already that both approaches, the traditional, the rational and the unbiased approach, both give you variants that provide very high titers of lysine. And similarly, we do have scenarios in both cases where the lysine titers drop. So we know we are having a real impact on the biology of the system that we are studying. We then took the top hits from all these libraries and rescreened them or retested them for confirming the lysine titers. And the question we were asking is, lysine biology is extremely well-studied system in E. coli. Given that, can we identify novel targets both in the rational approach and in the unbiased approach? And we were able to identify targets in both these cases using the scale and diversity of the edit types that we've put into these libraries. So the next slide, let's look at the pathway engineering results. As I said before, we targeted about 19 lysine biosynthesis pathway genes and about 20 transcription factors. These are targets that are very well studied, and we also identified key edits or in all these different enzymes that were already published in literature. So we were able to validate previously studied targets. In addition, we also identified a lot of new hits. In total, we had 38 unique hits. And when I say hits, what I mean is edits on a given target. So you would have the same target having multiple different edits that gives you an increase in lysine titer. And to give you an idea of the scale of increase that we see a fold improvement from anywhere from 2-fold to 5,000-fold over the wild type. This is fantastic because it helps us revalidate what we already know and identify new targets. But what about the unbiased approach? Do we really see hits in this case? We did see novel targets in this, and what was really interesting is most of what many of the hits that we identified were in the Y genes in E. coli. Even though E. coli is such a well-studied organism, a large part of the genome is called the Y-ome because the genes we do not understand the function of these genes in the E. coli genome. And it was extremely interesting that we identified several of these Y genes as targets in improving lysine production. So if you look at the graph here, what you see in the x-axis, especially in blue and green, are the different hits coming from the different library strategies. The green bars represent a genome-wide promoter insertion, a hit from a genome-wide promoter insertion library, and the blue bar indicates a hit from a genome-wide knockout library. There are two different backgrounds strains in which we built these constructs. The first was a wild type background, and the second was a positive control background where you have DAP-A, E84T is a mutant that creates the increase in lysines, because DAP-A is a very well-known enzyme in the lysine biosynthesis pathway, and this particular protein substitution relaxes the feedback inhibition by lysine and hence increases lysine production. What you can see here is that multiple hits have been identified both in the wild type background and in the positive control background that gives you very high increases over the background in which they have been edited. And predominantly, you find hits both in the Y genes, and we've also found a handful of hits in the amino acid biosynthesis genes, but this time in either the knockout or the promoter insertion scenario. While the previous slide, it was primarily saturation mutagenesis. So we established that the type of edits you make and the targets you choose and going genome-wide all have an advantage in identifying great novel hits. Now, for rapid forward engineering, it is important to combine these hits and keep moving up that phenotypic landscape. Our current platform delivers single edits into cells. We are working on a platform that is truly multiplex and combinatorial. But how do you use this current platform to combine hits? We started here. I'll give you an example where you can rapidly scale the phenotypic landscape even in this particular platform by combining the hits. In round one, as I told you before, we identified DAP-A variant, one of which was a protein substitution. And there was another one that gave a very high increase, which was a promoter, high expressing promoter inserted upstream of DAP-A. So we chose the DAP-A E84T strain as a background for a second round and rebuilt the libraries in that background and immediately identified a 10,000-fold improvement over wild type, where you had both DAP-A protein modification and an expression modification with the promoter insertion. For the next round, we used this particular strain as our background strain and built the libraries on them. And we saw a further increase in lysine production, where we had the DAP-A edits overlaid on stacked with the edit. For instance, in this case, what is highlighted is an edit in GlenD gene that is with a promoter insertion upstream of it that gave close to 14,000-fold increase over wild type. So stacking such edits is fairly common in practice in forward engineering. But what is remarkable here is the time frame in which we were able to achieve this. We were able to achieve 3 rounds of this process, getting to 14,000-fold increase over wild type in a span of 5 weeks with very limited FTE footprint. Most of our time was spent actually in screening for the lysine phenotype. So this example shows us that you can easily go multiple rounds to get to your phenotype of interest by combining complementary approaches of rational target identification and unbiased approach, and use a variety of edit types to move really fast and scale the phenotyping landscape. The second example that I want to walk through here is a typical genome discovery approach where we identified causal mutations in genes that impact cell growth. So typically, the previous example showed us how we singulated the isolates and we had isolate level screening. This particular example, which is a collaboration with Dr. Jeff Boeke at NYU, will highlight how we can use these libraries in a pooled selection scenario and use the power of the barcode sequencing to rapidly identify genotypes that drive the phenotype of interest. So the Sc2.0 project is a global collaboration to build a designer yeast genome from scratch. So what they are doing, these are labs across the world. They are designing, assembling, and characterizing synthetic versions of all 16 chromosomes of yeast. And they want to put it all together to create the first synthetic yeast containing all synthetic chromosomes. So our collaboration with Dr. Jeff Boeke centers around editing the Syn6 strain, which carries a synthetic version of chromosome 6. As part of building the Syn6 chromosome, several design elements have been added to the strain for different reasons. For example, they've created PCR tags to be used as watermarks to identify the engineered strain over the background. They've introduced LOXP sites to induce genome rearrangement, etc. At the end of this build, what they identified was that the Syn6 strain, because of repeated engineering, does accumulate intended and unintended changes and displayed growth defects. So this is very common. As strain engineers, you probably know that repeated engineering does cause strain on growth. And that is exactly what they saw in this case as well. Typically, to be able to deconvolute what causes this change is an extremely slow process. And in the case of Dr. Boeke's lab, what they identified took several approaches, like sequencing of the strain or identifying mutations through a meiotic recombination strategy, which is extremely cumbersome and laborious. Finally, they identified 4 causal mutations in 2 genes, MOB2 and PRE4. So the question was then posed to us as in, can the Inscripta genome engineering platform enable the identification of targets that rapidly reverses the growth defect? So instead of going through a long, cumbersome genetic process, can you use the Inscripta platform with pooled selections to get to the end identification of these targets, which you can then quickly revert in the strain engineering process? So the strategy we took was to be agnostic to any changes they had identified before. So it was almost like a Pepsi test. So we took all the designs that they had created to make that Syn6 strain and reverted them to wild type. So what you see in this table here are the different edit strategies for all the elements that they had inserted or all the elements that they had engineered and reverting it to a wild type. This was a great example because it is a mixture of edit type in a single library. While in the previous case, many of those libraries had a single edit type, like a saturation mutagenesis is all substitutions, which you saw in the lysine example. While in this example, we have substitutions, deletions, insertions, etc. in a single library. The total came out to around 485 designs, and what we designed was double that number, because what our software allows, and this is a very interesting feature of the software, is that you can create redundant designs. So it enables you to study design efficiency for any given targeted edit. You can compare between the 2 designs and ask if both of them worked or only one of them worked, etc. So we created around 939 designs. And the next step, I just want to show you how simplified the process is to identify the targets. It took us about a month to get to the library, where 3 weeks of that is getting the reagents to the platform, and making the library takes about 2 to 3 days. And once you get the library, what we did was run 3 biological replicates as pooled serial growth selections. and that was a week. So within a span of a month, we were able to go from making the library, running the phenotype, and then for each of these pools, we sequenced the barcode, which is an NGS sequencing assay, and we were able to analyze which of the barcodes enriched with respect to time zero or depleted with respect to time zero. That indicated to us which designs or edits were driving the phenotype such that the cells were getting a survival advantage and enriching in the population, and which of these designs had a detrimental impact and were falling out of the population. So we were able to study that population dynamics with respect to time zero across the different time points that we had run the experiment. So, and that within days you can identify that. So let's see what the data looks like. What you see here is a typical volcano plot, and on the x-axis is a log2 fold change of the barcode counts with respect to at time t with respect to time zero, and in this case it's a 24-hour time point. On the y-axis are the adjusted p-value, and if you take the quadrant, the rightmost quadrant, which shows very high fold change, which is greater than 4-fold change and highly statistically significant, the question that they were asking was, can we identify these targets that gave growth defects? And what was really interesting is we were able to recapitulate the edits that we reverted the phenotype of MOB2 and PRE4, and those edits actually were enriched in the population, which then says that by reverting the edit compared to what they had engineered, we were able to revert the phenotype, whereas MOB2 caused a growth defect in the engineered state, reverting the edit, reverting the genotype gave us an increase in survivability. And in terms of the barcode count itself, what was very stark here is at time zero, you can see the counts were about 1,000, and by 24 hours, you can see the increase in magnitude to about 20,000 barcode counts. So this was a great validation of how the Onyx platform can rapidly enable identification of genotypes driving the phenotype of interest. And what you see here are other designs also that enrich in this population. So the question we are asking now, not only have we recapitulated the MOB2 and PRE4, there are other designs here that do give a positive phenotype for survival. And it's extremely easy now to take that set and revalidate them as individual designs. So instead of going through months and months of experiments to identify that genotype to phenotype relationship and going stepwise, going gene by gene, we were able to, within a single month, identify a large set of targets which can then be taken into individual validation experiments. So to give you a quick summary of what we mean by rapid genome scale engineering with the Onyx platform, we spoke about scale, performance, and access. And let's look at the specifications around this. The Onyx platform has been developed right now for E. coli and Saccharomyces cerevisiae, and they have been optimized for K strains and B strains in E. coli, S288Z and SNPK in Saccharomyces. And customers can test both the standard strains and any related derivatives of these strains on the Onyx platform. In terms of the genome access, you can see from all the examples that I've shown today, you can access both the coding and non-coding regions in native genome and heterologous sequences. So if you've introduced heterologous sequences, you can access both the heterologous sequences and the native genome to scale your study of the biological system you're interested in. Now, we talked about scale. The designs per library indicated here shows the flexibility that you get in controlling the scale that you want to study. Both for E. coli and Saccharomyces, we start with a lowermost end of 100 designs per library. And for E. coli, you can build up to 10,000 member library, or for yeast, you can go up to 6,000 member library. And in terms of performance, we have precise edits, which is 1 edit per cell, with a diversity of edits such as insertions, deletions, substitutions, up to 63 base pairs across all of these edit types. And the library composition typically gives you greater than 90% designs covered and greater than 30% edited cells. And this is across all plexity, all library sizes. And the fraction of edited cells can range anywhere from 15% to 75%, primarily dictated by the targets you choose, of course, because if you choose targets that have fitness impacts, that will definitely reduce the number of edited cells in the library, because while we control the technology of editing, we don't control the biology. And ultimately, in terms of time to do an experiment, the design itself is extremely simple. The design user interface is just requires you to bring your targets and the type of edits you want to make. All the other choices around PAM sites and donor template lengths, etc., is optimized by the software. So anywhere from within minutes to about an hour, you can build multiple libraries, design multiple libraries. Once you order, the reagent delivery takes around 3 to 4 weeks, and the library build on the instrument at your benchtop takes about 2 to 3 days, which is 2 days for E. coli and 3 days for yeast. So what you see here is that your hands-on time in actually building these libraries is almost negligible. It is less than an hour to build these powerful libraries that can drive biology to accelerate your discoveries in your lab. So in summary, the Onyx Microbial Platform delivers a very user-friendly workflow for rapid cycles of learning. And you can use this to study either forward engineering experiments or genome biology. And what we would like to ask you now is, what can you do now that you have full access and potential of the genome without limiting your imagination? How would you like to use the Onyx platform? Thank you very much for your attention today. And I'd be very happy to take any questions.
Okay, awesome job. Well, we are all set pretty much from here. I'll just do some quick editing on the front and back end since there was no interruptions during your presentation. And then we'll get it ready for the event on Wednesday, and then I'll work offline with Natalie to situate the time slot for you.
Awesome. Thank you so much.
Okay. Well, we are all set. If anything comes up on your end, feel free to reach out. If not, we will go from here.
Okay, great.
Alrighty. Have a great rest of your day.
I'll just log out here, or I just close this, right, and that's it. Okay.
Yep, exactly.
Great. Thank you so much, Tyler. See you.
Thank you. Bye.